Hello, my name is Shanice Jones.
And I'm Kate Harden.
In this week's lab, we examined the respiratory, circulatory, and nervous system of the pig. It was also examined the amount of carbon dioxide in one's breath in comparison to room air concentration. Also, the effect of exercise on carbon dioxide production was observed, as well as we also measured the vital capacity within our section in our class.
The circulatory system transports a wide range of substances throughout the systems of the body. These substances include nutrients, gases, wastes, and hormones. Oxygen and carbon dioxide are two gases involved in this process. These gases are exchanged in the lungs as part of the pulmonary circulation circuit. The circulatory system is closely related to the respiratory system, which also deals with oxygen and carbon dioxide. The process of oxygenating the blood and removing carbon dioxide from the blood occurs because of the presence of the respiratory system.
The lab was completed according to the lab procedure titled Fetal Pig Dissection Part 3, Circulatory Respiratory Nervous System by Buchholz, along with the Dissection Guide and Atlas to the Fetal Pig by David G. Smith. In the pig dissection, the respiratory, circulatory, and nervous system were observed. Many organs were identified, such as the heart, lungs, and brain of the pig. In the slide, you can see the dissection of the heart.
Comparison of room air with exhaled air. 2 beakers were filled with 50 milliliters of lime water. In the first beaker, room air was bubbled into the lime water for 2 minutes, being continuously observed. In the second beaker, exhaled air was bubbled into the lime water for 30 seconds, also being continuously observed. After 2 minutes of bubbling room air through the first beaker, no change was observed. After only about 28 seconds of bubbling exhaled air through the second beaker, it was observed that the lime water had become cloudy. This suggests that exhaled air has a higher carbon dioxide content than room air. The calcium hydroxide in the lime water reacted with the carbon dioxide in the exhaled air to form calcium carbonate, turning the liquid from clear to cloudy. Effect of exercise on carbon dioxide production. 3 beakers were filled with 200 milliliters of phenolphthalein. Room air was bubbled into the first beaker until the phenolphthalein concentrate turned from pink to clear. Exhaled air was bubbled into the second beaker until the liquid turned clear. And in the third beaker, exhaled air after 2 minutes of vigorous exercise was bubbled through until the phenolphthalein turned clear. After the air had been bubbled through the beakers, ammonia solution was added dropwise to each beaker until the phenolphthalein became pink once again. The time it took for the phenolphthalein concentrate to turn clear was recorded, as was the number of drops of ammonia which turned the clear solution back to pink. Figure 1 shows the normalized values of the drops of ammonia solution required to turn the phenolphthalein pink, using the data collected by several lab groups. As made clear by the figure, exhaled air at rest has a greater carbon dioxide content than room air, and exhaled air after exercise contains an even greater concentration of carbon dioxide than exhaled air at rest. When comparing the average normalized number of drops of ammonia, exhaled air has 92% more carbon dioxide than room air. The exhaled air after exercise contains 35% more carbon dioxide than exhaled air at rest. These results were as predicted because carbon dioxide is the product expelled by animals when oxygen is taken in during cellular respiration. During vigorous exercise, the uptake of oxygen is greater than it is at rest, as cells require more energy to fuel the body. The heavy breathing that accompanies vigorous exercise leads to a greater release of carbon dioxide.
Pre-made slides were also viewed of the mammal peripheral nerve, a rat brain cerebrum, and a human lung under the microscope. These slides can be seen respectively. In the human lung slide, alveoli can be seen as well. When dissecting the pig, the intestines were removed. The intestines' length were determined to be 118.5 inches in length. This was the small and large intestines combined. The total length of the pig was also determined to be 18 inches in length. In comparison, the intestines were found to be 6.5 times longer than the actual pig. It is usually found that the small intestines is to be 5 times as long as the pig. The reason why our measurements were so much greater is because the length of the small and large intestines were taken together. Vital capacity of each group member was taken by exhaling forcefully into a spirometer. Those measurements were then compared to the class average. It was then found that group member 2's capacity did not fall within the normal curve. This is found by adding the standard deviation to the average. This could be due to the fact that the member was sick at the present time taking these measurements and were not able to breathe.